So we're going to start to talk about evolutionary change today, and in doing so we have to just go back and revisit a few of our terms that we would have covered in Biology 20, and that includes things like population and natural selection. So let's just take a look at population here very briefly. So from an ecological point of view, a population is defined as all members living in a certain area of the same species. And remember what a species is. By definition, a species is a population of organisms that can interbreed with one another. They're willing and able to reproduce, and most importantly, they can have viable offspring that can go on to reproduce as well. So a population consists of all members of a species that are living in the same space at the same time and who are capable, of course, of reproducing. So when we start to talk about members in a population, then we can start to talk about the different kinds of genes that they have within that population, and that very generally is called the gene pool. So the gene pool basically is the genes of all the members in a population that exist at one time. Evolution is changes in the frequency of those genes in that population. So if we see certain traits begin to become more dominant, or other traits begin to come less frequent, I should say, then we can have something that's called evolution, which means that the gene pool is actually changing. So let's take a look again at Darwinian theory of natural selection specifically. Remember, natural selection is basically when organisms. So natural selection is based on Darwinian theory, which basically means that those who are best suited to the environment are most likely to survive and most likely to reproduce and pass those desirable traits on to their offspring. And remember, with natural selection, it is the environment that is exerting pressure on organisms to adapt, not people. So when we talk about people deciding which individuals in a population get to breed, so if you think of like dog breeds or horse breeds, that is not natural selection, that is artificial selection. And because it's not the environment exerting the pressure, it is human beings deciding who gets to reproduce based on what characteristics the humans have decided are desirable. So natural selection, of course, will affect a gene pool in a population because the genes that are more favorable or provide a selective advantage for individuals that have those selective advantage genes, they're going to be more likely to survive and reproduce and pass those desirable traits on to their offspring. So one thing that we look at with evolutionary theory then and population genetics is we take a look at, okay, is there actually a way that we can follow genes through a population and see how the frequency of those genes is actually changing? So that takes us to our next piece on population genetics and finding those gene frequencies within a population. So by definition, gene frequency is exactly what the name implies. It's the frequency of that gene in a population, or in other words, how often that gene will appear in that population. And again, remember, a population is a group of individuals that live in the same area at the same time, and of course can reproduce and have viable offspring. So population sampling then is done because it's very difficult to actually count every member of a population, particularly if we're dealing with populations that are quite large or with species that are difficult to actually gain quantitative data on. And as a result, something called population sampling is done. So that's kind of akin to in class me asking, hey, how many people here can actually roll their tongue? Because that's a gene, and we're going to see if we can identify the frequency of that gene in our population. So if we kind of assume that our population is the classroom and our 30 plus students, and then I say, okay, who's a tongue roller here? And individuals that actually are capable of that raise their hands, then we actually have a population sample. And then what we can do is we can say, okay, 13 out of 30 people in our classroom actually were capable of tongue rolling, so now we're going to extrapolate that to the larger population. And so that's what population sampling is for. It's basically taking a small sample of the population, determining that gene frequency or how often that gene is appearing in that population, and then kind of projecting that value to a larger population. So as we said, for example, the tongue rolling versus the non-tongue rolling. So we can do this with virtually any genetic trait. So if we take a look at a gene pool a little more specifically, as we said, basically a gene pool presents the values of how many of specific genes are present in a given population during a certain time. And certain populations can have high gene frequencies for certain traits and low gene frequencies for other traits. It just depends on the population, how large the population is, whether or not individuals are moving into or leaving that population, and other factors as well. So scientists will use these gene frequencies to determine if there are actually changes or evolution happening in a population. So just to look at the graphics here, we can see we have some wild boar, and then we kind of have one trait here identified with our allele combination here. So we have, let's just say that this is coat color. We'll just throw that out there. And so we can see that we have a number of individuals that are homozygous recessive. We have a number of individuals that are homozygous dominant, and then of course we have our heterozygous individuals that have one of each allele type. So basically what we would do to determine allele frequency is we would separate those genotypes into the individual alleles, and then we would say that those are the allele frequencies in that particular gene pool for that trait. So for example, our individual here that is homozygous dominant, we would say, okay, this individual has 2 dominant alleles, so 2 dominant alleles go into that gene pool to determine frequency. Our little heterozygous individual here, this individual has 1 dominant allele, 1 recessive allele, so we separate those, and 1 more dominant allele goes into our gene pool, and 1 recessive allele goes into our gene pool to calculate that gene frequency. So literally, when we talk about gene frequencies, we're saying how many of each type of allele, dominant versus recessive, is actually in that particular population. Now, as far as studies on humans go, it's a little difficult to study gene frequencies in humans for a number of different reasons. And one of the biggest reasons for that is that we, compared to other species like fruit flies, for example, or other insects, rodents, etc., we produce relatively few offspring, and so the generations take a really long time to actually come up with a number of individuals in a population that could be studied, okay? So it makes it difficult as far as human studies go, and that's why quite often when it comes to calculating gene frequencies, other species that have more prolific rates of reproduction are used as opposed to human beings. There's also ethical considerations when it comes to studying human beings as well. There has to be consent and those types of things too. And observing evolution over time, or observing those gene frequency changes over time, takes a really, really long time to do. And so when you have generations that live for, 30, 40, 50, 60, 80 years, it's really hard to actually track those over a short period of time. And that's why species like Drosophila that can reproduce, say, every 14 days, they are used for these types of studies. Because of the fact that it's very easy to trace those gene frequencies over multiple generations in a very short period of time. A lot of human traits are also affected by the environment, okay? So when we look at things like, a person's weight, a person's height, whether a person has an autoimmune disorder, all of those things can be very much affected by environment as well as genes. So sometimes it's hard to determine whether or not the allele frequencies are being altered because of environmental things or selection or what. Okay, so there definitely presents some unique challenges when it comes to those studies on humans. So where does the Hardy-Weinberg principle come into all of this? Well, in 1908, Hardy and Weinberg weren't working together actually. They were working independently, but they both kind of derived the same basic principle and concerning population genetics, and it is called the Hardy-Weinberg principle. And the Hardy-Weinberg principle basically states that the frequencies of alleles in a population's gene pool is going to remain constant over generations if, and here's the big if, all other factors stay constant. And we call this genetic equilibrium. So when we do not have a change in the allele frequencies over many, many generations, okay, so those numbers, the number of recessive alleles and dominant alleles stay the same in a population for all traits over time, we call that genetic equilibrium. Now, this isn't something that will happen in the natural world because according for the Hardy-Weinberg equilibrium to actually hold fast, it has to be an unchanging gene pool, and that's something that is very difficult to do in the real world. So when Hardy and Weinberg were both coming up with their principle, they basically ignored the factors that influence or can influence populations, all right? And those things include the population must be very large, okay? So we have to have a huge population, not just a very small population like with an endangered species of, 100 or 200 individuals. So we have to have a very large population. There can be no emigration or immigration into the population. So you can't have any members leaving, you can't have any members of the population coming in, new members of the population coming in. And if you think about that, that kind of makes sense, because if your new people are coming in or organisms are coming in and other individuals are leaving, then they're taking their genes with them, or they're bringing new genes into the gene pool, and that's going to alter that equilibrium. So nobody in, nobody out. The other thing is no mutations, okay? So that means that as mitosis occurs, as meiosis occurs, there's literally no mutation to create new alleles and add those alleles to that gene pool. Random mating. So this basically means that mating is a free-for-all. There's no sexual selection. So you know how we talk about how certain individuals, when they are looking for a mate, they select for certain characteristics. They battle with each other to determine who has the rights to mate with a group of females, for example. None of that happens. This is just random mating. You basically stumble upon a mate and you mate. So there's no sexual selection occurring at all. And the last criteria that has to be in place for Hardy-Weinberg equilibrium, which remember again means that there's no change in the gene pool over many successive generations, is that there is no natural selection. So this basically means no one has a selective advantage. So all traits are equal in terms of their advantage to the individual that has those traits, and nobody has any kind of selective advantage. So the environment can exert all the pressure that it wants, but there isn't going to be any natural selection based on who is best suited to the environment, who's most likely to survive, okay? So all traits equally aid in survival and equally detract in survival. So there's no preference given to any individual based on their particular characteristics. Now, obviously, these things don't occur in any kind of population, really. It doesn't matter if it's a natural population or if it's a population like in a zoo or anything like that, or even with artificial selection. There's really, no such thing as all of these things kind of staying in place. But remember, Hardy and Weinberg purposely ignored all of these factors that can affect gene pools to establish what genetic equilibrium is, okay, in a specific population. So these 5 points for Hardy-Weinberg equilibrium are very important, so you need to make sure that you understand those for sure. So then we can kind of go on to the mathematical piece. So how do we actually use Hardy-Weinberg equilibrium? And remember, this means that, over time, if equilibrium is in place, genetic equilibrium means that there are no changes in the allele frequencies in a population, so they stay constant over successive generations, all right? So what can we actually use Hardy-Weinberg equilibrium for? We can use it to determine genotypic frequencies in a population, and we can use it to determine if evolution is actually occurring. So think about that for a minute. What are the genotypic frequencies in a population? So how many homozygous, dominant, heterozygous, and homozygous recessive individuals do we actually have in a population? And then, depending on what happens with our numbers, we can actually determine if evolution is occurring or if the population is in genetic equilibrium. Okay, so here's our equation for Hardy-Weinberg equilibrium. So p squared plus 2pq plus q squared is equal to 1, okay? And then p plus q is always equal to 1. And that piece is a really important one to remember. So no matter what your value of p is, your value of q when added to that has to be 1, and vice versa. No matter what your value for q is, your value of p when added to that has to be equal to 1. Okay, so what do these different variables mean in this equation? So p, and notice that's not p squared, p represents the frequency of the dominant allele. And so if we're going to use that, just the letter A for our genotype combinations, notice that this is a dominant allele, not a genotype. So P represents the frequency of all the dominant alleles in the population. So that would be our capital A's, right? Q represents the frequency of the recessive alleles in our population. So that would be our lowercase a, right? Now P squared is the frequency of our homozygous dominant genotypes, because A times A, right, is A squared. So P squared represents the homozygous dominant genotypes. Q squared is the homozygous recessive genotypes. And lastly, 2pq is the frequency of our heterozygous individuals. Okay, so we kind of have two things going on here. The P and the Q relate to the alleles specifically. So how many dominant alleles, how many recessive alleles in a population for a certain trait. And then P squared is how many homozygous dominant individuals do we have. Q squared, how many homozygous recessive individuals do we have in the population, and 2pq, how many heterozygous individuals, so one dominant allele, one recessive allele, do we have in the population. Okay, so we can just take a look at a little example here with our little insects here, okay? So we kind of have our little, looks like little insects here, yeah, because there's 6 legs here. So here's our little insects, and we're going to just take a look at how the Hardy-Weinberg equation can apply to this, okay? So if we kind of look down here to our insects, we have a homozygous dominant individual here, and you'll notice that individual has the black color. And then we have our heterozygous individual, who has the black color as well. So clearly this is a completely dominant trait. And then we have our little homozygous recessive individual who is like white or gray, whatever color you want to say that is, okay? So now we kind of know what we're dealing with, right? In terms of our numbers. So remember what our Hardy-Weinberg equation tells us. So remember, P is the frequency of our dominant alleles, Q is the frequency of our recessive alleles, P squared is the frequency of our homozygous dominant guys, 2pq is the frequency of our heterozygous little guys, and Q squared is the frequency of our homozygous recessive little guys, or our little white guys, okay? So if we take a look at this, it kind of gives us a little bit of information here. And it says that P is equal to 0.3, so this is information that we were given, and Q is equal to 0.7. So here's what we have to remember. We have to remember that the frequency of P is equal to our dominant alleles, and the frequency of Q, that is our number of recessive alleles, or frequency of recessive alleles, right? So if we remember, our P plus Q has to be equal to 1. This does indeed equal 1, right? Because 0.3 plus 0.7 equals 1. And then what we can do is we can find out P squared and Q squared, right? So if P is equal to 0.3, then P squared is equal to 0.3 squared, which is 0.09. And then we can do the same thing with Q, right? So then if we figure out Q squared, we can go 0.7 times 0.7, or 0.7 squared, and then we get our value for Q squared. And remember what that is, kiddos. P squared rather is the number of homozygous dominant individuals in a population. Q squared is our number of homozygous recessive individuals in a population. And then for our frequency of our heterozygous individuals, remember that we use 2pq for that. So notice this isn't squared, this is 2pq. So 2 times 0.3 times 0.7 is 0.42. Okay, so what does that actually mean in terms of our genotypic frequencies in the population? 9% of our population is homozygous dominant, 42% of our population is heterozygous, and 49% of our population is homozygous recessive. So that's how that's one example of how the Hardy-Weinberg equation can be applied. So let's just take a look at another example here. We'll just work through a couple of examples so that we know what we need to do in terms of our calculations. So it says, suppose a certain allele A, okay, so here's your dominant allele A, has a frequency of 0.6 in a population. Okay, so let's think about what that is. Allele A has a frequency of 0.6. So I want you to think about, is this our Q, is this our P, is this P squared, what is this, okay? And then it says the frequency of the allele, the other allele, must be 0.4, because remember, P plus Q is equal to 1, right? So if one of those values is 0.6, the other one has to be the difference between 0.6 and 1, which is 0.4, right? So we have 1 minus 0.6 is equal to 0.4. So that's how we got our other value. So let's see what happens during reproduction, and we can actually take a look at how we use the Hardy-Weinberg for this. Okay, so if we are looking for our homozygous dominant individuals in a population, then of course we're going to use p squared. Now we already established up here that 0.6, because it's the frequency of the dominant allele, that's going to be p. And so p squared is equal to 0.36. So of course what that means is 36% of the population is homozygous dominant. If we look at our 2pq, remember, that's what gives us our heterozygous individuals. So 2 times 0.6 times 0.4, that means 48% of the individuals in the population are heterozygous for that trait. And then of course lastly, we established over here that q is equal to 0.4, okay, because we went 1 minus p, which was 0.6 equals 0.4. So q squared, which represents our homozygous recessive individuals, q squared is 0.4 squared, which is 0.16. So that means, of course, that 16% of the individuals in the population are homozygous recessive. So just another example of how we can apply that Hardy-Weinberg equilibrium. So one more example just to do together. This one's a little bit different because you'll see that it gives us a percentage as opposed to the actual P or Q value here, right? So it says a dominant phenotype is seen in 38% of the population. Okay, now let's pay attention here because it says a dominant phenotype, okay? It doesn't say a dominant genotype, it says a dominant phenotype. And listen, when we're doing these Hardy-Weinberg equations, we always assume complete dominance unless we're told otherwise, okay? What percentage of the population would be expected to be heterozygous for this trait? And remember, when we're talking about our heterozygous individuals, that is our 2pq. All right, so let's break down what we actually have here. So what we were given here is the phenotype, all right? So q squared is equal to 62% or 0.62, and therefore q is equal to 0.79. Now sometimes kids will be like, where the heck did this 62% come from? Well, remember that if we're saying that 38% of the population has a dominant phenotype, then 1 minus 0.38 is going to give us our q, okay? So there we go. And then q is equal to 0.79 and p is equal to 0.21. And that, of course, is because once we get our value for q, which is 0.79, we can subtract 1 from that, and we get 0.21 for p. 2pq, remember, is how we find out our heterozygous individuals in the population. And so 2 times 0.21, which is p, and then times 0.79, which is q, and that gives us 0.33. So obviously that means that 33% of the population will be heterozygous for that particular trait.